Hey guys, this is Kendall Terry, and this is DNA transcription, going from DNA to RNA. In essence, we're looking at how genes make proteins. Well, understand that we first have to go from DNA that's inside the nucleus, and the DNA that's inside the nucleus is too big to fit out of the nuclear pores, so it can't get out into the cytoplasm. Now, the importance in that is because in the cytoplasm is where the ribosomes are, and the ribosomes, you'll see in the next lecture, is what takes the nucleic acid code and makes proteins. So DNA can't get to where the ribosomes are, so it has to make a messenger to take that code. The messenger that it makes is called RNA. So RNA is a nucleic acid, very similar to DNA, except for three main differences. The first is that RNA consists of a single strand of nucleotides instead of the two strands found in DNA. So when you look at the picture here, this is RNA, only one strand. This is DNA, and you can see the two strands that are wound around each other. The next is that RNA nucleotides contain a 5-carbon sugar called ribose, as opposed to DNA that contains deoxyribose. It's really one oxygen molecule that makes the difference. And although that's a small difference, it's still a difference nonetheless. The third thing is that RNA contains the nucleotides adenine, the nitrogen bases, adenine, guanine, and cytosine, but it does not have thymine. Instead, it has a base called uracil. You can see it right here in the picture, uracil. Now, uracil acts just like thymine. In fact, it's also one of the pyrimidines. It's where we get the cutting the pie. Cytosine, uracil, and thymine are all pyrimidines. Uracil acts just like thymine, but it's what is in RNA, whereas thymine is not. Thymine is in DNA. So a gene's instructions for making proteins are coded in the sequence of nucleotides in the gene. You can think of it as kind of like our alphabet, and depending on how you arrange those letters of the alphabet, you will get a sentence. And if we take those same letters and we rearrange them, then you get a different sentence. It means something else. Well, in DNA, they only have 4 letters to choose from: A, T, C, and G. And the order of those letters will depend on the code for making a particular protein. So that code then, the instruction for making the proteins, are transferred from the gene in the DNA to a messenger RNA molecule. This is called mRNA, in a process called transcription. It's transcribing the message from DNA to RNA. Cells then use two different types of RNA called, that we put together, that we just say is a ribosome, and we'll talk about ribosomes more in the next lecture. And they will use that to make a sequence of amino acids based on the instructions in the mRNA. And that is called translation, and that'll be the focus of the next lecture. Let's look at the process of transcription, though. Transcription begins when RNA polymerase, that's the enzyme, RNA polymerase binds to the gene's promoter. Now, a promoter region is simply a region that gets RNA polymerase's attention. You can think of it maybe as similar to like a commercial. If you're watching TV and there's a commercial that comes on TV that gets your attention, that you think, I could, go and have that cheeseburger, or I really need to drive that car, or I may need that new iPhone, right? So that's a promoter in essence. It's getting your attention to say, hey, you need our product. Well, in DNA, genes also have promoters. It gets RNA polymerase's attention and says, hey, there's a gene here that needs to make a protein. You need to bind here. So RNA polymerase will bind there, and it will start. This promoter will act as a start signal for transcription. So it binds RNA, that RNA polymerase. That is the first step, and you can see that here. See, RNA polymerase is kind of like this blue bubble. The second step then is RNA polymerase, same enzyme, will unwind and unzip the DNA. Now, in replication, DNA helicase did this, but in transcription, it's RNA polymerase that does all this. So it will unwind and separate the two DNA strands, exposing those nucleotides. This is the important region, that sequence of nitrogen bases. If you know this sequence, then you can figure out the next steps of the process. So RNA polymerase, when it has exposed these bases, RNA polymerase will then add the correct RNA nucleotides. So it kind of, what we say, reads the gene, and as it reads it, it will attach the correct RNA polymerase or RNA nucleotides to the DNA. These do not stay attached, though. These will come off of the DNA and be a single strand out here that will leave the nucleus. This follows simple base pairing rules, except whenever there's an adenine on the DNA, it can't attach a thymine to it because there's no thymine RNA. Instead, it will attach a uracil in that place. And that will make the RNA molecule that will then leave the nucleus and go out into the cytoplasm where the protein will be made.